We'd gotten as far as talking about the parasympathetic effects on the heart rate. And what we're going to do today is, and we've gotten as far as beginning to discuss the sympathetic effects, and we'll try to finish that off today. And I'll quickly go through the sympathetic effects on the conductile cells. And I got to that in more detail in this section than I did in the second. We'll talk about the conductile cells and how in the conductile cells, the sympathetic innervation, that is the post-ganglionic sympathetic neurons that are releasing norepinephrine. The norepinephrine, by binding to a Gprotein-coupled receptor, specifically a Gprotein-coupled receptor that has a three-dimensional shape on it that will bind norepinephrine. The binding of the norepinephrine to the Gprotein-coupled receptor leads to the increase of cyclic AMP by stimulating adenylyl cyclase. We talked about that last time in terms of the HCN channels, because HCN stands for hyperpolarization activated cyclic nucleotide gated. So by increasing the amount of cyclic nucleotide, cyclic AMP, we increase the rate of opening the HCN channels and thereby increase the rate of rise of the ramp potential. And that's what we finished off with yesterday on the conductile cells. And we emphasized a bit yesterday that the parasympathetic branch of the autonomic nervous system, the post-ganglionic parasympathetic innervation, is primarily to the pacemaker cells in the SA and the AV node, and to a lesser extent in the bundle branches. And that there isn't very much innervation of the contractile cells by the parasympathetic branch. So most of the autonomic effect on the heart is coming from the parasympathetic innervation of the pacemaker, and then sympathetic innervation to the pacemaker cells, but also to the contractile cells. So one of the things I want to emphasize here is that unlike the parasympathetic branch, whose innervation is predominantly to the pacemaker cells, to the conductile system, the sympathetic branch now can innervate the sympathetic branch now can innervate, in addition to the conductile cells, innervates the contractile cells. Much of the same story pertains, that is, the binding of neurotransmitter to the Gprotein-coupled receptor increases cyclic AMP. But now there's the follow-on of the old cyclic A story that we told. That is, increasing cyclic A activates protein kinase A, which phosphorylates a whole raft of proteins. The raft of proteins in these contractile cells that are phosphorylated are the dihydropyridine receptors, the RIAND receptors, and the calcium pumps. And so this is going to lead to an increased release of calcium from the internal stores, because we'll phosphorylate the DHP receptors, allow more calcium to come in. We have more calcium binding to RIAND receptors, more calcium comes out through the RIAND receptors. And then because of the increased cytoplasmic concentrations of calcium, and not only that, we've also increased the heart rate because of the sympathetic effect, now we also have to start advocating for the calcium pumps. Calcium pumps now have to play a role in removing the calcium more rapidly, and we'll talk a little bit about that. Then we can talk a little bit about the effects on the contractile cells by the sympathetic branch of the autonomic nervous system on the pressure volume loop. And again, if you can see this, you understand the cardiac cycle, and you understand how increased contractility in the contractile cardiomyocytes will squeeze more blood out of the chambers and lead to a lower end systolic volume. And so we'll talk about that in terms of the PV loop. Then we'll take another step forward and talk about cardiac muscle contraction and electrocardiogram. These are these images you've seen on any number of emergency room shows. And what the electrocardiogram is, it's a measurement of the massive movement of ions, of current, into and out of the contractile cells. The conductile cells are virtually unrecognizable in the electrocardiogram. Those conductile cell potentials are simply swamped by the tremendous amount of ionic movement into and out of the contractile machinery. The first one we'll see is the P wave. The P wave will be atrial contraction. So we're looking at the ionic movement, mostly of sodium, into the atrial cardiomyocytes. Followed then by something known as the QRS complex. So remember, there's a slight delay between the P wave the slight delay between the atrial contraction and the ventricular contraction. The QRS complex represents ventricular contraction. And then finally, there's this thing called a T wave. And you'll notice this is just letters in the alphabet, P, Q, R, S, and T. T wave is potassium efflux due to the repolarization of the ventricular, mostly ventricular cardiomyocytes. Now, we'll go through that fairly quickly. If you talk to a trained cardiologist, they can look at these electrocardiograms and read remarkable things about what's normal and what's abnormal in a heartbeat. And we're just going to go very quickly through them, trying to emphasize how these are measured, what we mean by each of these waves, and how the ions are moving for each of these waves. Then I'd like to finish with a discussion of the vasculature. We've really touched on this idea of the vasculature, but we emphasized the ideas of the vasculature mostly through the coronary artery. And everything we talked about in the coronary artery is going to be true here as well. But I want to bring this round to the fate of the blood once it goes out into the systemic vasculature. And of course, the route of the blood, remember, this is always a closed system. The blood never leaves the tubes. So it goes from an artery to an arteriole to a capillary to a venule to a vein back to the right side of the heart. That's always the pattern. Then what we're going to talk about is that the arterioles, particularly, as well as the arteries, in addition to their lining of endothelial cells, these cells are also wrapped or surrounded by a layer of smooth muscle. So the contraction of the smooth muscle now can affect the radius of these tubes. And you'll remember from Poiseuille's law, it doesn't take much in the way of a reduction or even an expansion in the radius of the tube before we get an enormous effect on the flow. So what we'll see here is that we can regulate the movement of blood through the tubes, through particularly the arterioles, by using the alpha subunits, the alpha-1 receptors rather, of the norepinephrine Gproteincoupled receptor to now cause smooth muscle contraction, and then by affecting the contraction of the smooth muscle, we can now guide blood from one place to another, depending on which ones are contracted and which ones aren't. And that largely results from Poiseuille's law and the enormous effect, the exponential effect on R to the fourth. But let's start here with a quick discussion, and I'll show this in green, of sympathetic effects on pacemaker cells. So this box is intended to represent a pacemaker cell. It's part of the conductile system. And up here in blue, I'm going to try to represent the output regions. This is meant to be the output of the sympathetic post-ganglionic neuron. There's nothing fancy about this. These output regions, just like we've seen a million times before, contain synaptic vesicles. And the synaptic vesicles now are filled with the neurotransmitter, norepinephrine. When the action potential arrives at the output region here and opens the calcium channels, that now in turn works its way through the T snare and D snare complex. But in the end, we release these molecules of norepinephrine. And here, as transmembrane proteins, is a type of Gprotein coupled receptor. Now, I should mention here that these G-protein coupled receptors are a family of receptors. Something like 4% of the genome is dedicated to producing this family of proteins. And we have a great variety of these. We have some of these G-protein coupled receptors that bind norepinephrine. Others we saw yesterday bind acetylcholine. If we get into the central nervous system or even in parts of the peripheral nervous system, we find that they'll bind neuropeptides. So they have a vast array of molecules they can bind to. And each one of them is a different G-protein coupled receptor. The one we're talking about here is the G-protein coupled receptor that has a specific extracellular three-dimensional shape which permits it to bind the neurotransmitter norepinephrine. Once the norepinephrine is bound to the extracellular region of this transmembrane protein, it has an effect on the cytoplasmic side of the protein. We commonly show this as someplace near the carboxy terminus, but it's probably due to a rearrangement of several portions of the cytoplasmic part of the protein. And the rearrangement then allows for the alpha subunit of the trimeric. This is a trimeric GTP binding protein. This trimeric GTP binding protein only binds GTP to the alpha subunit. Typically, the alpha subunit, though, when it's associated with the beta and gamma, has on the alpha subunit a GDP. But when this alpha subunit has access to the open site here on the G-protein coupled receptor that's already bound to norepinephrine, when that happens, then we exchange the GDP for GTP. The happy association of the alpha, the beta, and the gamma subunits now come apart. And the alpha subunit, with its GTP attached, goes off and binds to one of these transmembrane proteins. It has like 24 transmembrane proteins, which is in fact 24 transmembrane regions, which is adenylyl cyclase. The role of the adenylyl cyclase is to convert ATP into cyclic AMP. So the binding of norepinephrine to this G-protein coupled receptor through its interaction with the GTP binding protein leads to an increase in activity of the adenylyl cyclase so that inside the cell we get an increase in concentration of cyclic AMP. That cyclic AMP now can interact with the subunits. There are 4 subunits of the HCN channel. And the way we left this was to say that this HCN channel has a hole in the middle. It's a proper pore. It's a cation channel. It will let sodium come in. But there are 4 subunits, and each of the 4 subunits has a site where we can bind cyclic AMP. The more of these 4 sites we have bound to cyclic AMP, the greater the probability this channel swings open and lets sodium come in. And if you remember, the big deal from yesterday was that when we talked about the pacemaker potentials, there's this slow depolarization, opening of voltage-gated calcium channels, repolarization by potassium channels, and a slow depolarization all over again. And this region is a depolarization that results from sodium influx through HCN channels. So by increasing the amount of cyclic AMP here through the effects of norepinephrine on these conductile cells, by increasing the number of cyclic AMP molecules, we increase the probability of these channels opening. So we effectively can change the slope of this depolarization. We can effectively change the slope of the ramp potential. So by increasing the slope, in other words, by making the slope of this potential steeper, we more quickly reach threshold and fire another action potential. So we have the effect of norepinephrine increasing the heart rate by bringing the pacemaker potentials more rapidly to threshold to fire an action potential. Yes?
I noticed, I can't remember if you told us, there only needs to be one cyclic AMP bound to HCN for sodium factor, right?
So the question here is, what's the relationship between cyclic AMP binding and the probability of channel opening? And I've skated right by this. And the reason I skated by it is that one will open it, but it's a low probability. Binding the second increases that probability. I suspect this is a cooperative event, but I don't know. I haven't looked at the kinetics of this. But I'm pretty sure that it's a cooperative event. So with none bound, you probably won't open it at all. With one bound, it's a low probability, but it's not twice as likely when you bind the second. It's probably more like 5 or 10 times. It may be logarithmic, maybe 10 times the probability. That's known. I just don't know it. There's a real number for that, and it's known. Right? Yes.
Is there I guess a limit for like how shallow it can be or how steep it can be? Is there like a limit to it?
So the question is there a limit to the steepness of this ramp potential? And not being too glib, I'd say yes, and the limit is death. And if you get this thing too long, then your heart rates are so slow that you have a real problem. But one of the things we haven't discussed, and it's a really important issue, if you get these heart rates too high, once you get heart rates up above, even in the young, when you get your heart rates up above about 200 or 240, particularly 240, then there's simply insufficient time for the ventricles to fill to provide an adequate volume of blood in the ventricle to be pumped out. And so you need a certain interbeat interval to allow for diastolic filling. And if this gets too steep, then this becomes fibrillation. And then you simply don't have adequate time for diastolic filling to do its job. So the quick answer is you better get it right so you don't die. But in fact, there's a broad range over which that's functional.
I guess what's the question? The question would be like, what's the lower limit? Like
How low can you go? Depends on how quiet you are, I suspect. I don't know the real answer to that. I know that it's not unusual for well-trained athletes to have heart rates 42, 43. I don't know of many that go below that. And I'm go way out on a limb here and say that I know that surgeons get very twitchy when they start having someone come into the surgical suite with heart rates in the low 40s. Because they're afraid they're going to add some level of anesthetic and lower that heart rate even farther, and then they're going to put someone in crisis. So there is a lower limit. If you ask a cardiologist, you'd get a real answer. But I really don't know where that lower limit is. But I suspect it's in the 30s someplace. Anything else? Not. So that's the story for the conductile cells. Now let's redraw this for the contractile cells. So in red here, this red box is meant now to represent a contractile cell. Now, in contrast to the parasympathetic branch, which innervates predominantly, overwhelmingly predominantly, the conductile system, the sympathetic branch has innervation to both the conductile system, i.e. the pacemakers, and these contractile cells. Now, remember one of the major differences between the conductile cells and the contractile cells, as the name implies, is these contractile cells contain sarcomeres. And you'll remember from the discussion earlier in the week that a characteristic of these cells is that they contain T-tubules. And along with the T-tubules, right next door to the T-tubules, is this well-developed, well-characterized structure of the sarcoplasmic reticulum. The major function, the job of these contractile cells, of course, is to generate force. These are the business end of the heart. This is where we're going to generate force both in the atrial myocardium and in the ventricular myocardium. So these muscle cells, these contractile cardiomyocytes, play the role of having their myosin heads reach up, contact the open sites in the actin, and draw together the Z lines to generate force. That's their job. The rate at which we see the contraction in these cells is largely dependent upon what's going on up here in the conductile cells and the rate of rise of these pacemaker potentials. But it turns out that there's also a fairly robust innervation here of these contractile cells of the beta-1 norepinephrine receptor. The beta-1 norepinephrine receptor is a type of Gprotein-coupled receptor. And in fact, this beta-1 norepinephrine receptor is the same one that we labeled up here. I just didn't label this Gprotein-coupled receptor as a beta-1 receptor, but it's still a beta-1 receptor, the same ones. Now, what we're going to see from this point out is similar to what we see in the conductile cell, but we're going to add one caveat to it. Let me put this receptor over here. And now there is the trimeric GTP binding protein, alpha, beta, and gamma. The alpha subunit normally has the GTP on it, but when it interacts with a Gprotein-coupled receptor, one of these beta-1 receptors, when the beta-1 receptor has bound to it norepinephrine, we go through the same process above. That is, we release the alpha subunit from its hold on the beta-gamma. The alpha subunit here comes loose with its GTP attached. And it, just as above, interacts with the purple adenylyl cyclase. And the same story pertains. That is, we end up, by activating, turning on the adenylyl cyclase, we end up increasing the cyclic AMP concentration. The main story in the conductile cell, however, is that the cyclic AMP itself plays a role. The cyclic AMP itself is basically a cytoplasmic neurotransmitter or a transmitter at least, which binds to the HCN channel, increasing sodium influx. Now what we see here in the conductile cells is an additional effect. Cyclic AMP activates protein kinase A. Whenever you see this term kinase, this tells you it's an enzyme that phosphorylates It's an enzyme that phosphorylates proteins. Now, any time that you covalently link a phosphate group to a protein, and in this case, it's going to be to serines and threonines, but that's a detail. Anytime you put this great bulky phosphate group on with all its charge under a protein, it changes its function. And among the proteins that are going to be phosphorylated by the protein kinase A, it's oftenly abbreviated pKa, phosphorylates DHP receptors and ryanodine receptors and calcium pumps. Let's take these two first. You'll remember that the dihydropyridine receptors are calcium channels. They're voltage-gated calcium channels in the T-tubule membrane. So these are the DHP receptors. And the calcium that comes through the DHP receptors then affects these ryanodine receptors. And by the calcium coming through the DHP receptors binding to the RYR receptors, we now increase calcium release from the internal storage compartment. We talked about that the other day. So now what's happening is we're increasing both the influx of calcium through the DHP receptors by phosphorylation of the DHP receptors. These are the voltage-gated calcium channels, remember, that increase in calcium influx leads to increase in activation of ryanodine receptors to increase calcium release. So all of that is driving the system to put more calcium out in the cytoplasm. Now, from what you know about muscle contraction, you can see what's going to happen. By having a larger amount of calcium, greater calcium concentrations out here, particularly with respect to the thin filaments, there's going to be more calcium bound to the troponin C. That's going to have more of those troponin Cs, causing more tropomyosins to move. That's going to expose more expose more myosin binding sites on the actin. So now we have a greater number of these myosin heads that can interact with the actin, and we're basically going to get a stronger and more rapid contraction. So what we see here is that all in all, this increase in phosphorylation increased phosphorylation of the calcium channels, both the dihydropyridine receptors and the ryanodine receptors, increases the strength of muscle contraction. So that's going to generate more force. And you had a question.
You kind of answered it there, but so it's not the influx of calcium that opens the ryanodine receptors?
It is the influx of calcium that opens the ryanodine receptors.
So what is the protein kinase A doing to it?
So what does the protein kinase A do? The protein kinase A acts as an enzyme which attaches phosphate groups to the dihydropyridine receptors and the ryanodine receptors. When these proteins become phosphorylated, it changes both the probability of opening and also changes, I believe, the amount of calcium that can move through them. So you get both a change, so you'll get more of these channels open and the calcium permeability of those channels becomes greater.
So they just kind of work together to release calcium?
So the two work together. The dihydropyridine receptor is the voltage-gated calcium channel, which initially allows calcium to come in. And then that calcium binds to the ryanodine receptors and initiates the process of calcium-induced calcium release. And that releases the calcium from the sarcoplasmic reticulum, binding then to the troponin C. Anything else? Put your phone away, I'd appreciate it. Yes?
Do you also say that it increases the rate that it contracts to, or is it the strength?
It will be both the rate and the strength of contraction. You look perplexed.
I just don't understand how, but I guess more inflexible.
So when we talk about the rate, I'm simply talking about the rate at which these Z lines are being drawn together. So not necessarily the heart rate. Let's not confuse it with heart rate. I'm just saying the rate at which these myosin heads will move up and contact. You have simply greater number of open myosin binding sites. You have more of them, then you'll cycle them more quickly and bring this together more quickly. Right. Now, there's a problem here, and I think this is what you're alluding to. The problem is that if we're going to have an effect on the pacemaker cells and increase the rate at which these action potentials are arriving through the conductile system to the contractile system, if that's getting faster and our heart's beating faster, that means we have, and we're also releasing more calcium to increase the strength of contraction, then that implies we better get rid of that calcium faster because we're going to have residual calcium here that is going to keep the myosin binding sites open. So one of the other effects here is the phosphorylation of the calcium pumps. So what the calcium pumps will do is they'll pump calcium more rapidly back into the sarcoplasmic reticulum, so you can start the process all over again. So it's a very beautiful system where in the process of increasing the rate of heartbeat, as we see from the conductile cells, and therefore we increase the amount of calcium in the cytoplasm, both because of the increased rate and because of the effects on the DHP and RYR receptors. We also simultaneously, concomitantly affect the pumps so we can clear that calcium more effectively. Any other questions? Let's now just take a minute and try to put all of this into the pressure volume loop. Let's take the right-hand side of this panel and talk again a little bit about the pressure volume loop. It never hurts to talk about the pressure volume loop because if you can see this pressure volume loop and what's going on in each case and walk your way through an understanding of the pressure volume loop, you have a pretty good understanding of what's happening with the heartbeat itself. So we'll label this as the pressure volume loop, also known as the toast diagram. And you remember we started here at point A. We had the period of diastolic filling. Right at the end there's a little bump going to point B. We said that this little bump was an increase in pressure. It represented atrial contraction. That provided the last squirt of blood into the ventricle. And then during the period of isovolumetric contraction, the pressure increases, reaches a point where the pressure inside the ventricle is greater than the pressure outside, and the valve opens. Comes over to here, the valve closes, and comes back down again. And we said between the lines A and B and C and D, we had this thing known as the stroke volume. Now let's imagine on this what's going to happen if we increase the contraction on the upper panel. If we increase the contraction of those contractile cardiomyocytes. So we basically put a greater squeeze during the period of isovolumetric contraction. What will happen is there's now going to be generating a greater force. The greater force probably won't have too much effect on the opening of the aorta, but now we see a greater force of pressure coming out, and because of the greater contraction, we're now going to force more blood out of the ventricles. But then, during the period of isovolumetric relaxation, we come straight back down again. And now we can label these as A prime, B prime, C prime, and D prime. And if you look at this, what we've done is we've now increased the stroke volume. We haven't affected the end diastolic volume, that's just filling. So all we've done is allow it to fill. We've had no effect in this greater contraction, stronger contraction on diastolic filling. The effect is on the pressure generated squirting the blood out into the aorta. And that contraction, because it's stronger, will squeeze more blood out of the ventricle. And so now, instead of having a typical end systolic volume, instead of having a typical end systolic volume of, say, 70 mils, we can now drive this to the left so it's around 50 mils. So we're increasing the stroke volume by increasing the ability of these contractile cardiomyocytes to give a harder squeeze and pump more blood out. Any questions? So what we've done here so far, let me show you a couple of slides. What we've done so far is to walk our way through the autonomic nervous system and the effect on the heart. So let's take a minute here and review what we've been doing. This first image is just a prettier picture of the three-box model, showing on the left the central nervous system where we have relegated the neuronal cell bodies to the preganglionic neurons. The preganglionic neurons is the one will synapse onto the postganglionic neurons out here in the autonomic ganglion. Then the postganglionic neurons are fated to innervate smooth muscle, gland, or cardiac muscle. In our story here today, that was both the innervation of the innervation of both the conductile and contractile cardiomyocytes. Then there's the comparison of that which is sympathetic and that which is parasympathetic, and that's what's shown on this slide. And it emphasizes the idea that acetylcholine is always the neurotransmitter between the preganglionic neuron and the postganglionic neuron in both the sympathetic and the parasympathetic branch. Always acetylcholine. The acetylcholine binds to nicotinic receptors in the postsynaptic cell, generating an EPSP that will produce action potentials, leading to the release of neurotransmitter onto the target. So again, this is just a recapitulation of what we had on the earlier slide. Preganglionic to postganglionic to target. Preganglionic to postganglionic acetylcholine, nicotinic receptors. Postganglionic onto target now is a different neurotransmitter. Postganglionics for the sympathetic branch release the neurotransmitter norepinephrine, whereas the postganglionics for the parasympathetic branch release the molecule acetylcholine. And from that point on, we can have different types of Gprotein receptors. And what we'll see here is that first we have this difference in the anatomy, where this is showing the motor nucleus of 10. It also shows some of the other cranial nerves. We emphasize the motor nucleus of 10, which now had its preganglionic parasympathetic neurons going out into the periphery, contained within that structure called the vagus nerve or cranial nerve 10. The vagus nerve, being the wanderer, goes throughout the thoracic and abdominal cavities and innervates all manner of organs in those cavities. And what this is showing is that preganglionic neuron synapsing onto a postganglionic neuron on the heart, and that postganglionic neuron having a short axonal process that innervates the target itself. That's in contrast to what we see here in the sympathetic branch. In the sympathetic branch, what we see is the preganglionic sympathetic neurons located for sure in the central nervous system, but now in the spinal cord, T1 to L3. Those preganglionic sympathetic neurons send processes out into the periphery, and in the periphery they synapse onto postganglionic neurons. Now, because these sympathetic chain ganglia are right next door to the vertebral column, the axons of the preganglionic neurons tend to be pretty short. They synapse onto the postganglionic sympathetics in the sympathetic chain ganglia, and those postganglionic neurons then send these great long processes. Some of them go up into the head, and the ones we're interested in, though, go to the heart. So now we have these two systems, the parasympathetic branch and the sympathetic branch, innervating the heart. Then they have differential effects on the heart itself. But this is a nice figure because what it's showing is it's showing the difference in the anatomy along with the difference in the innervation pattern within the heart itself. And I'll walk you through it. Here are the parasympathetic preganglionics, whose neuronal cell bodies would be in the motor nucleus 10 of the brainstem, coming all the way down here into the cardiac ganglia. The cardiac ganglia are associated with the sinoatrial node and the atrioventricular node. The preganglionic neurons then synapse onto the postganglionic neurons. This is going to be the release of neurotransmitter acetylcholine onto the postganglionic neuron, eliciting the action potentials now to release acetylcholine onto the SA node and the AV node. That acetylcholine now is going to bind to Gprotein-coupled receptors whose effects are to reduce the rate of rise of the ramp potential. On the other side here is the sympathetic innervation, and this is meant to be describing the innervation of the the innervation of the postganglionics by the preganglionics. So this would represent the sympathetic chain ganglia. Preganglionic to postganglionic, release acetylcholine, same process, nicotinic acetylcholine receptors. The action potentials down here now are releasing the molecule norepinephrine. This is showing that it's releasing norepinephrine onto the pacemaker cells of the AV node, the SA node, and to some extent the bundle branches. But in addition to the innervation of the conductile system, here with the sympathetic branch, this is showing the innervation of the ventricular myocytes, cardiomyocytes. And this is the effect we just showed. We just showed the effect of the sympathetic branch on the degree of contraction here in the ventricular cardiomyocytes. And this is simply showing the idea of the Gprotein-coupled receptors. There are a gazillion different Gprotein-coupled receptors, each one binding to a particular neurohormone, binding in this case to acetylcholine or to norepinephrine, and each of those are different. They bind out here in the outside, causing a structural change, allowing for the alpha beta gamma subunits to interact with the cytoplasmic side of the Gprotein coupled receptor. When that happens, the alpha subunit goes over and talks to adenylyl cyclase, upregulating adenylyl cyclase activity to increase cyclic AMP concentrations. In the conductal cells, that cyclic AMP acts directly on the HCN channels. But in the contractile cells, there's an additional set of effects, and that is the cyclic AMP activates protein kinase A to phosphorylate a whole bunch of things, not the least of which are the voltage-gated calcium channels, the DHP receptors, the ryanodine receptors, as well as those calcium pumps that are involved in removing the calcium. And that's the summary of everything we've gone through today. So, any questions to there? If not, let me take a minute quickly and draw on the board to the back panel. And draw on the board here something known as the electrocardiogram. What the electrocardiogram is measuring, it measures the flow of sodium ions into cardiomyocytes. I should emphasize here specifically contractile cardiomyocytes, and the flow of potassium ions out of the cardiomyocytes, the contractile myocytes. Now, this is a fairly old technique. This was developed in the early 1920s, and it was developed well before we were measuring the kinds of intracellular ion movements we've talked about in here. The measurements of the electrocardiogram are measurements from a series of electrodes placed on something like 14 different parts of the body. And if you've ever had an electrocardiogram, you know they put some electrodes on your chest, they put some on your fingertips, they put some on your toes. There are many different electrodes. And what they're effectively doing is they're measuring the movement of these ionic currents into and out of the cardiomyocytes. And because of the position of these extracellular electrodes, they're measuring the movement of lots and lots of ions into and out of the heart. And when we do that, we get something that we can graph in terms of time. And I'm going to show this as voltage, but we could equally call it current. And what we see is that there is this hump, which is known as the P wave. Oh, let me move this up so I have a little more room. There's this P wave, followed by a downward shift, an upward shift, and then a downward shift, then back to baseline. This set here is known as the QRS complex, where this is the Q wave, this is the R wave, and this downward deflection is the S wave, followed by something known as the T wave. Now, what this actually represents in terms of the ionic movements that we've described in here is this P wave here represents sodium influx into atrial cardiomyocytes. The QRS complex shows sodium influx into ventricular cardiomyocytes. These are both depolarizing events. And then finally, the T wave represents the efflux of potassium from ventricular cardiomyocytes. In the last minute, let me take, I shouldn't have buried it. Let's look at this. So, as you're looking at the PV loop, where is this P wave? It's right at it's that little hump at the end of diastolic filling. What's the QRS wave? The QRS wave is the contraction of the cardiomyocytes, because it's influx of sodium into the cardiomyocytes and a little bit of calcium, and that's going on throughout the period of isovolumetric contraction and well into the ejection phase where the blood's going out into the aorta. And then during the period of isovolumetric relaxation, the potassium now is flowing out. And so this is when the potassium is flowing out of the ventricular cardiomyocytes. And some of you may well ask, when you look at this, where is the efflux of potassium from the atrial cardiomyocytes? That's buried in the QRS wave. Let's leave it with that, and we'll come back next time and talk a little bit about the vasculature.